Receptor tyrosine kinases, another type of membrane receptor proteins. This tyrosine kinase receptor protein has a neat shape. Inside or intracellular of the cell, we have a polypeptide that has tyrosines, which tyrosine is a type of amino acid along the protein. On the outside of the cell, we have a signal binding site. This is where the signaling molecule will attach. Now, the name receptor tyrosine kinase comes from the fact that this receptor protein has kinase activity. A kinase is simply an enzyme that catalyzes the transfer of phosphate groups. So you can kind of guess that throughout the reception process, that phosphate groups will be attaching to somewhere. And in this case, we will be attaching phosphate groups to the different tyrosines. So therefore the name tyrosine kinases, because kinase means attaching or transferring phosphate groups, and we'll be transferring them to tyrosines. So here's how it works. Here we have a signaling molecule will attach to the signal binding site, and this will cause a conformational change. In this case, the two monomers or polypeptides will aggregate or come together. When they come together, they will form a dimer. Now, we have not yet added the phosphate groups, so this is called an unphosphorylated dimer. However, the tyrosine kinase regions are activated at this point and ready to transfer phosphate groups. So, we'll get those phosphate groups from ATP molecules. We will be taking a phosphate off of ATP, which leaves us with a DP and inorganic phosphate, and that phosphate will attach to a tyrosine. Now, it's the kinase activity that is responsible for transferring that phosphate group to the tyrosine amino acids. Now, this will happen enough times so that all tyrosines have phosphate groups. This molecule is now phosphorylated. We've added phosphate groups. So it's called a phosphorylated dimer. It is a fully activated tyrosine kinase. Now, the next step, if the whole purpose of these signaling molecules is to cause a response inside the cell, we need to change their message into a cellular message. So the next step would to have inactive relay proteins come up to the molecule and attach to a phosphorylated tyrosine. So when this relay protein is bound to this molecule, it causes a conformational or structural change in the relay protein. Now, this change will lead to a series of events or a signal transduction pathway, which leads to a cellular response. Now, it's kind of cool that this one molecule has many phosphate groups, and in other molecules you may even have more, leading to maybe 10 different cellular responses. So, another example, we have another relay molecule might come and attach to a very specific phosphorylated tyrosine, leading to a different cellular response. So again, you could have up to 10 different cellular responses, all from one signal binding event. This is different than a G protein, which a G protein would just have one cellular response.